Our first speaker today is Anoushka Joglekar from Weill Cornell Medicine. Anoushka received her degree in applied mathematics and English from The Ohio State University. And currently, Anoushka is a PhD candidate in the Tri-Institutional Computational Biology and Medicine program at Weill Cornell Medicine. She works in the neurogenetics department in the Tilgner lab, where they focus on alternative splicing signatures in the brain. Anoushka was attracted to this field because she tends to get very excited when people push the boundaries of what is known in science with cool new technology and with math that seems to work like magic. Outside of science, she enjoys the theater and all things literary and occasionally indulges in circus arts. Today, she will be speaking about her recent work published in Nature Communications titled A Spatially Resolved Brain Region and Cell Type Specific Isoform Atlas of the Postnatal Mouse Brain. So, welcome, Anoushka.
Thanks, Nikki. And thank you for the introduction. As you could see, my title is kind of a mouthful, but before I jump into any of my talk, I like to usually start with a nice little example. So I'm sure many of you recognize this cartoon of C. elegans, and one fun fact about C. elegans is that it has about 20,000 genes in its genome. And the reason why this is a fun fact is because humans also have about 20,000 genes in their genome. And yet I would argue that we're maybe slightly a little bit more complex than the worm. So of course there's many reasons for this to happen, but one of the primary, I would argue, reasons is alternative splicing. And so I don't need to tell you about the central dogma where you go from DNA being transcribed into RNA, which is then translated into protein. But an often overlooked aspect of it is the process of RNA splicing, where you get rid of introns that fall between the exons of your gene, but sometimes you also get your exons being taken out, which leads to multiple proteins which have distinct function arising from the same pre-mRNA molecule. And these proteins are called protein isoforms, and their precursors are therefore called mRNA isoforms. And so worms actually have maybe about one isoform per gene, whereas humans have about 10 on average. So we have 200,000 proteins as opposed to the worms with their lowly 20,000 proteins. So there's different types of alternative splicing, and we don't really have to go into the details, but the most common one in mammals is exon skipping. But then you also have types where your entire gene starts later or ends earlier, which is alternative first exons or alternative transcription start sites. You'll hear me say TSS quite a bit, and alternative last exons or alternative polyase sites. And the reason why this is important is because it has massive implications in health and disease. So I'm sure many of you recognize the CD33 gene. It's an immune gene, and it's present in the microglia in the brain, and it's known to be implicated in Alzheimer's. There's two exons in here that encode different protein domains, and there's some isoforms which skip the exon 2 or the VSET domain, leading to a neuroprotective role for the CD33 gene. So it literally, the smallest change can lead to massive implications in life and death, or just quality of life in general. Now, you know that many genes are expressed in different parts of the body, but sometimes you'll have different organs having different alternative splice forms, and they can then cause a full cascade. So there's like organ-specific splicing, but if we zoom in, you can have a lot of brain region-specific splicing. So the same gene being expressed in two different brain regions can have different isoforms being expressed. And then if we want to zoom in even a little bit further, there's different cell types within the brain that have different isoforms being expressed. And what's the best way to study anything at a cell type level? So you have droplet-based single-cell RNA-seq, which like revolutionized the field when it came out a few years ago. So the way this works is you have a microfluidic device wherein you have barcoded beads that you send in along with your cells of interest from whatever tissue, as well as oil. And each cell gets trapped within a droplet, And once your cell gets lysed, every molecule gets a little barcode. So you can trace back exactly which cell each molecule came from, which is great. However, the caveat is that it's a 3' quantification method. So what that means is that you will get your barcode information per molecule, but you'll only get part of your actual gene. So if we go back to our original cartoon, you'll maybe be able to distinguish between some proteins, but all you get is that this is gene A and that is the count for it, but you can't really distinguish between protein A and B. So obviously we needed to find a way to get around it, and therefore our lab came up with a technique called Scissor Seek, or single-cell isoform RNA sequencing. The first half is the same as I just previously described with the short read aspect of it, where you then process your cells through whatever single-cell pipeline and assign them to different cell types. However, we reserve part of our cDNA and send it off for long-read sequencing, and we've been using PacBio for it, where you get full-length information for the same molecules that you sequenced on the short-read sequencer. What that means is that you can basically get barcodes from those molecules that correspond to your short-read barcodes and assign them to cell types, therefore getting spliced forms or isoforms of your different cell types. Obviously, it never looks as clean as this, but that was the idea. And we also wanted to quantify this. So the way we did that was we looked at the P7 or postnatal day 7 mouse across 2 brain regions. The PFC, which is the prefrontal cortex, it is useful in a whole bunch of cognitive tasks, as well as the hippocampus, which many of you know is implicated in memory. And what we wanted to ask was mainly 2 questions. So we know there's region-specific splicing, but the question is, how does it arise? So for the same cell types that are present across the 2 brain regions, do you see splicing differences? And the second part was that for a single brain region, in this case, the hippocampus, because we sequenced it super deep, 4 different cell types within that region, do you see splicing differences? So before we get into the results, I wanted to quickly go over how we quantify these splicing differences. So consider this hypothetical gene where you have 2 alternative exons flanking 2 constitutive exons, which means that if you were trying to sequence it on an Illumina machine, you wouldn't really get the connectivity information. So historically, like 10 years ago, what people used to do was calculate PSI, or percent spliced in, where you look at the percent of number of reads for an exon, so the red exon, over the total number of reads for that gene. So you get like a PSI value of 0.5, which is great, but you don't really know if the red and blue exon occur together or not. And the other caveat to this method is that because you're testing this on a per exon basis, you can have a lot of different exons per gene, right? And so when you're statistically, when you're trying to correct for multiple testing, you end up using a very harsh correction. So instead, now that we have PacBio sequencing and we have this full-length information, what we did was we just called it percent isoform, where we look at the number of reads for an isoform divided by the total number of reads, which comes out to be like a 1/4 for each isoform. So you really get the connectivity information. And on top of that, for every single gene, you have 1 test. So you have an n by 2 table, and therefore when you're correcting for multiple testing, you can use a more kind correction for multiple testing. So with all of this and the pipeline used to detect the barcodes and the long reads and whatnot, we created this package called Scissor Seeker, and a friend of mine created this logo. And this is what I use for the majority of my analysis. So the first question, like I said, was trying to find the between region variability in isoform expression. And like I was saying, that you can have differences that you see on a broad, like pseudo bulk level between hippocampus and PFC, but you don't really know where it's coming from. So there's 3 different models that this can arise from. So let's assume I'm just going to show a cute little cartoon as to how this can happen. So say you have hippocampal neurons and PFC neurons, which have an abundance of blue isoforms in the hip and an abundance of orange isoforms in the PFC. So the question is, where did this discrepancy come from? So let's say each region has excitatory and inhibitory neurons. So there's one model that says that both cell types can change splicing. So what that means is you can have excitatory neurons showing a lot of blue and going to excitatory neurons that show more red. Or inhibitory neurons showing a little bit of blue to showing none at all. So this is where both cell types change their splicing profile, leading to this region-specific splicing. The other type is where only one cell type changes splicing. So in this case, you can see this real stark difference between excitatory neurons, and the inhibitory neurons look basically the same across both brain regions. And the third one is kind of counterintuitive, and it has nothing to do with splicing at all. It just has to do with gene expression. So you could have a lot of the, so you could have excitatory neurons having blue, inhibitory neurons having red, but what changes is the degree to which they are expressing or transcribing these transcripts. So we wanted to see how much of the genome follows which model. So the way we first did that was by doing a very standard volcano plot where we looked at the delta pi, or the change in the percent inclusion of the isoforms, on the x-axis, and the FDR, or the corrected p-value on the y-axis, and we see a lot of genes that come up to be differentially expressed. And then we wanted to, I'm just going to illustrate with examples, but we wanted to see what kinds of models were followed. So one example is the NSFL1C gene, which I'm going to show you a plot where every line here is a transcript. This is the Gencode annotation. And then following that is every single line here is a transcript coming from the PFC, and this is the oligodendrocytes. And you can see that you see one isoform that is majorly expressed with some degree of intron retention, which could be an artifact or could be real, but I'm not really commenting on that right now. And in other cell types in the PFC, we see a very similar trend, that you have this one major isoform being expressed. What you see in the hippocampus, however, is that it's not that isoform that is majorly expressed. You have this one very small, I think it's like 9 nucleotides of a microexon that is expressed across all of the different cell types in the hippocampus. So it really is like a microenvironment thing where the same gene expressed in 2 different regions has a whole different splicing profile. And microexons are thought to be expressed in neurons, but clearly not. So this was a cool thing. So this model we see in about 20% of the cases, not too often. The second model, where only one cell type drives the splicing changes, is seen in the majority of the cases, so 70%. So this is a gene called hexa, and only one known isoform was annotated for this. However, we find that in the inhibitory neurons for HIP and PFC, it looks about the same, but if you look at the excitatory neurons, the PFC, like 70% of them really skip that exon, and that transcript has been known to be involved in nonsense-mediated decay, so it has a regulatory role in the prefrontal cortex that it does not have in the PFC. And then the third model, I'm not really going to go into because it was expressed in a very small percentage of genes. Now I'm going to come to the second part of my talk, and I'm going to try and wrap up really quickly. What we wanted to see was the within region variability. So we kind of did the same thing as I showed before, where we compared 2 different cell types. And we probed these differential isoform expression at every level. So at a broad cell type level, where you're comparing neurons versus everything else, obviously the neurons are very different. You have a pretty large percentage of genes that turn out to be differentially spliced. However, we found one surprising result, and that was that, although the neurons are different, even at the granular level, there's this one cell type called the choroid plexus epithelial cells. And they are super different, not just from the neurons, but also themselves, like within non-neurons. So when you sort of do this density plot of the percent of tested genes that are differentially spliced, you see that the within neuron curve is pretty broad. However, when you take out the choroid plexus comparisons as opposed to the others, you see a very clear shift. And then when we further probed it to see what was causing this, was it like the exons or the polya sites or the TSS, we found that it was largely the TSS that was mediating this. And one thing to note is, again, the TSS is on the 5' end, so this is not the information we could have captured if we were doing any sort of short read or regular 10x sequencing. And when, like, just to go a little bit further, we found that this choroid plexus, which is responsible for secreting CSF, prefers an upstream start site as opposed to the regular downstream start site, which is, again, crazy because it's like a large proportion of genes that are expressed everywhere have a preference for a different TSS in the choroid plexus. And so that's all for a broad cell type level on in non-neurons, but we also wanted to look at neurons because, like, that's getting at the heart of cell type specificity. So we looked at a gene called FGF13, which is expressed very highly at my time point, which is a P7 time point, and it's like developmentally regulated, and if you knock it out, it's lethal. So it's super important. And cell type specificity of isoform expression was not known for this gene. So there's two isoforms, again, mediated by a transcription start site. And we find that the excitatory neurons express a lot of the S isoform, a lot being almost close to 100%. And the inhibitory neurons express some bits of the Y isoform. And so we did, or a friend of mine did, some overexpression studies to see if this was functionally relevant. And what we found was that the S isoform is colocalized to the nucleus or the nucleolus, and the Y isoform is sort of everywhere. And we sort of took it a step further and validated with Basescope. So the way Basescope works is you basically design probes that target certain parts of your gene, and you can see exactly where it stands. So for the excitatory neurons, we use NeuroD6, that's going to show up in green, and then the two isoforms are both going to show up in pink. So what we found is that NeuroD6 is found in the CA region. Again, not surprising because it's an excitatory marker. However, when you zoom in, you can find that, like, the VY isoform doesn't co-localize at all. So the green and pink are super separate, versus over here, the green and pink are like on top of each other. So we did the same with GAT2, which is an inhibitory marker, and we find, again, that the S isoform doesn't co-localize, so green and pink separate over here, but on top of each other here, meaning that it really is cell type specific, and there's functional changes because you see them in different parts of the cell. So I'm going to take 1 minute to talk about the spatial aspect of it. So, like, so far we were dealing with one gene, or like the choroid plexus, which is like spatially separated. So this is like the theoretical spatial separation, right? Because we took different sections of our brain. However, it would be sweet if we could, like, take it all together. So 10x Genomics came up with their Visium platform, which is basically the same as single cell sequencing, except you have a slide and you can, like, mount your tissue on it, and it goes. I don't think that's a scientific term for it, but basically you can barcode every single spot on the slide, and therefore you can trace back where every single molecule came from. So obviously we had to take it and call it ScissorSeek because we sequenced it on the PacBio and Nanopore platforms, and we found not cell type specific but region specific isoforms, and that is like the future. So unfortunately, the resolution of these Visium slides is not super high, so there's going to be a lot of different cells in one spot, but, like, pretty cool technology, right? So one thing we found was that you could use the choroid plexus as proxy for non-neurons, and the hippocampus as proxy for neurons. And well-known cases of neuronal and non-neuronal splicing differences are super evident in these regions. And then lastly, one of my favorite results from the paper is this, again, this gene that is known to be differentially spliced, except you see this gradient of back to front where only the front expresses one isoform and only the back expresses the other isoform. And we saw this in a few more cases, but there's a lot more to be done in this area. So with that, I'm going to conclude my talk. Alternative splicing is cell type and region specific. There's one model, like the one cell type model, which is the most prevalent. cell type specific isoform expression is functionally relevant, so like pay attention to isoforms. And then finally, we introduced a new method for spatially resolving isoform expression, and it's got like super exciting things coming in the future. And I'd like to thank my lab and Anna and Susan, who did a lot of my imaging experiments. Some people over at Rockefeller and neighboring labs that helped out with sequencing, and then 10x Genomics and PacBio. Both, like multiple people from there, helped me out during the course of my project, and I'm very grateful for them. Thank you.
That was great. For some of the follow-up experiments you were talking about, is PacBio going to be used in any of those?
Yeah, so actually you know how I said we were doing the one time point, or what I did was the one time point in 2 brain regions. We've sort of expanded that to track developmentally as well as across different brain regions. And we're in the process of collecting data for that as we speak. And yeah, so we're using primarily PacBio for that, although it's going to be not as deep as what we did here. So we're doing somewhere between 3 and 5 smart cells for each sample, but we have like 20 samples. So that should be pretty exciting.
Nice. Yeah, that is exciting. I was going to say, I know that we're in the process of making improvements to that protocol, but I'm not sure when it'll come out. So I
Yeah,
was
I
going to say,
know.
hopefully it was going to provide you with some benefits, but it sounds like your work is wrapping up.
A little bit, yeah. So like I spoke to Liz Singh a while ago, like maybe like a month or 2 ago, and she was telling me about that. That's the one where you increase throughput by ligation, right? Yeah, so she said that you'd probably get the protocol out in a little bit, but I don't know if we have the capacity to sort of like sit around optimizing it because we've already started most of the sequencing.
Yeah, that makes sense. So I do see people starting to join in chat. So, I would just say feel free to ask Anoushka questions as you have them. So she's here to really answer any questions about her work or provide you with any insight onto the experimentation that she did. So, otherwise, two of us will just chat a little bit about her work. And so, again, feel free to ask questions as you have them. But yeah, so I guess one question that I had for you, Anoushka, too, when I was listening to you is, like, what do your splicing profiles look like at the single cell level? Like, can you give a little more insight into that?
Yeah, so unfortunately, we don't have that much depth, right? Because like short read sequencing, you're gonna get millions, like hundreds of millions of reads, whereas here you get somewhere between 2 and 4. However, there are a few genes for which you get like more than 50 cells, for which you get more than 20 to 40 reads per cell. And there you find like if there's a neuron versus non-neuron separation, we found this really cool gene where the non-neurons have very low inclusion of some isoform, and all of the cells in that cluster together at like 1, and then the neurons cluster together at -1 or 0, whatever the scale is. And then there's like these intermediate cell types, which is why like developmental time points are so cool, because they're sort of making their way into being neurons. And they were super spread out between the 0 and 1. So it's like depending on their identity, they either looked like individual cells, either looked like non-neurons or neurons. So hopefully at some point we'll get some genes which are like that, and we can probe further at single cell levels.
Yeah, awesome. So another question that came in is, do isoforms define your fine-grained cell states and are these different from the cell types?
That's a great question. So when we worked with PacBio actually last year or 2 years ago, they sequenced a bunch of smart cells for us for the hippocampus. And we wanted to see if we could like cluster in isoforms the same way you cluster on like short read sequencing to see if you get these cell states. Unfortunately, again, it's a depth issue, I think, more than an isoform issue, because you recover the cell types broadly, but not like a new cell type, which is what we were hoping for.
Yeah, great. And like I said, for anybody that just joined us, if you have any questions for Anoushka, related to her work or even anything outside of that, feel free to pop it in the room chat. And a question I have for you too, Anoushka, is I know you presented a lot on the 10x platform. Did you guys
Mhm.
try out any single cell applications or any other single cell platforms?
Like, you mean other than 10x?
Correct.
Yes, no, we did not. Because I mean, so the DropSeek I think is the cheapest to work with, but it's kind of gnarly to set up in your own lab space. So in one of the labs I rotated in before Huggins, they sort of had the whole like microfluidic setup, but it's not super reliable, so we haven't really done that.
And I've heard people say that they can see a lot of like 5' to 5' artifacts from the 10x system. Is that
Mm-hmm.
something that you guys noticed as well with your
Oh,
experimentation? And if so, how did you kind of like adjust for that?
yeah, so we call them like TSO artifacts because the TSO is on the 5', right? So we now have a working model where we can sort of get rid of them. And I don't know if I'm allowed to tell you exactly how we do it, but we're in the process of submitting a paper like in the next couple of weeks, and that sort of gets at that like artifact removal, and therefore you get a lot more of your usable transcripts.
Awesome. Yeah, that's good to know. Is your lab open to like taking in outside samples if other people are interested in doing that type of work?
I think so. I mean, I'll have to talk to my PI about it, but absolutely. Or like as soon as it's up on bioRxiv, I think it's like easy enough to replicate. It's not rocket science, but it's one of those things where people are afraid of competition. But yeah, if someone needs a sample to be run, then we will be happy to do it.
Great. And another question that just came in for you too is, could single cell Isoseek be applied to the Visium data too? And how does this compare to single cell?
Yeah. Okay, so first question, the Scissor Seeker, like the R package that we made, does apply to Visium. So that's the package I did use in like I don't know, the 2 slides that I showed. And we actually also like the entire pipeline can be used to the Visium data. And we did some clustering on those isoforms and got the brain regions pretty well separated out too. So for sure, there's just like a slight layer of putting it on top of the image, in terms of the coordinates, but that's not super difficult to do. And it compares pretty well to single cell. So like the only problem is the resolution. So you have about 5 to 10 cells per spot. And therefore you can't really tell that a cell is necessarily, say, a pyramidal excitatory neuron as opposed to an oligodendrocyte. But if you've used like the Seurat deconvolution methods, or there's others, you can sort of tell like 90% of this spot is X cell type. So you can sort of do these linear deconvolution techniques. So it's similar to single cell in terms of the processing. In terms of the interpretation, it's a little bit more difficult. And with SNRNA sequencing, so like nuclear sequencing, that is also something we've played around with. And it's applicable again in terms of the code itself. It works. It's just that you'll have a lot of intronic reads. So if you can sort of get rid of those intronic reads, then it will be a much better experience, I think. But other than that, I like in terms of the software, it's applicable.
Great. So a follow-up to that is, have you ever done scRNA-seq and Visium integration? And then any recommendations of tools that you use for that integration?
Yeah, so if you could clarify what you mean by integration, because so in terms of that deconvolution, I did do that. So like we sequenced the Visium slide the way I showed you on that on like at the end of my talk, and then we used the P7 like single cell data to figure out what each of the spots was. I wish I had like slides right now to show you, but yeah, so like you can use those same exact data sets, even though it won't be the exact same cDNA. You can sort of extract the profiles of the cell types. And then in terms of tools, I've mostly used the Seurat one. And then there's a one called Spotlight. So it's like an NMF regression model. Um, Yeah, same sample generated, yeah, across the single cell and Visium to get the cell type. So yeah, I would recommend the Seurat tutorial as well as the Spotlight because that seemed pretty cool.
Yeah, great. And they did clarify a little bit. So they were saying, yes, the same sample generated both um, the single-seek RNA-seq and the Visium to help deconvolute of the Visium spot cell type. And then another question here is, what is the fraction of barcodes you recover from long reads compared to the single-cell RNA-seq data? And then have you tried something similar with the ONP technologies?
Okay, I'm guessing that's ONT and it was a typo, but yes, we have done something similar with ONT. We generally just find that it's slightly lower quality, right? Because that's what you expect. And we did like a pretty comprehensive analysis of how things differ between ONT and PacBio, but that's a topic for another time. In terms of the fraction of barcodes, so like every single barcode that is assigned to a cell type in the short read analysis, like through the Seurat or whatever pipelines that you're confident in is a cell, every single one of those barcodes is recovered in the long reads. Of course, for some like cell types, the number of reads being assigned to that cell is going to be lower. So for example, appendable cells or like vascular leptomeningeal cells, like I don't want to say you don't care about them, but not a lot of people care about them. So you get not too many reads for that cell, but all of those barcodes are recovered. Just to clarify a tad further, we find barcoded reads in about like, so 40 to 50% of all reads have a barcode, if that was your question. Also, hi, Carlos.
Yeah, great. So, these questions are awesome. Keep them coming if you have them. You definitely have Anoushka's time here for a few more minutes. And I guess to another one that I had thought of when we were discussing, oh, 1 second. I think she just dropped off live.
Sorry, I think I don't know what happened.
No worries. So I was going to say, how does the cell type nature of splicing change in adult mice?
Oh, well, so we have some theories about it, and we don't fully know yet. So this is like the project I was alluding to before. We're in the process of collecting the data. But like my hypothesis is that like how you have like 40% of things that are different between say neurons and astrocytes. I think those percentages are going to change with age. And also the models that I was talking about, I feel like they're no longer going to be the exact same in terms of like one cell type changes between two brain regions or two cell types do. Like I think those are going to be dramatically different as you go into adulthood. But as of now, that's more of a speculative answer.
Yeah, great. Thank you. So yeah, that's all the questions I'm seeing right now. So, again, oh, I think one more did come in. So did you recover previously unannotated isoforms that were diluted over the bulk tissues?
Yes. So so the thing is that Gencode or whatever annotation that you use is by no means comprehensive, right? So like we always do recover a certain percentage, like it goes anywhere between 5 and I want to say 5 and 10. I was going to say 30. But what we did was when we're comparing, say, cell type A versus cell type B, and we find the isoforms that are the most important or the most switched between the two cell types, we isolated those. There were about like 75 that were like super relevant for the cell type specific splicing. And we had some collaborators over at Gencode, and we had them like manually go through the list and annotate them according to their biotypes. So yeah, we do find a lot of those that wouldn't be found in bulk because they'd be so lowly expressed.
Yeah, great. And then could you take your combined short and long read data and ask if you only had the short read data, could you tell what the dominant isoform is with some level of confidence?
Okay, that's a good question, and it's also something that so far, like until longer data was this high throughput, that is kind of what people used to do, like combine both and make sure that the splice sites were recovered in the short read data. But the problem with all of that is that even though you can qualitatively tell what an isoform or what the different types of isoforms are, the quantitative measure for that is a little bit difficult because you're doing some level of probabilistic modeling as to whether this read is coming from isoform A or B. That being said, for some genes, of course, you can do that, especially when it has a easy splicing profile. But then there's some genes with like 70 or so like alternative exons, and all of them are like a massive hairball. And those are very difficult to deconvolve using only short read data.
Great. Well, Anoushka, thank you so much for your time and your talk today. It was great, and good luck with your future projects.
Thank you.
If anybody else wants to connect with Anoushka, these recordings will be available. So as long as you sign up for the webinar today, which I'm assuming you have because you made it here, you will get a recording, and feel free to contact her or her lab with any additional questions that you have. So yeah, Anoushka, again, apologies.
Sorry.
Yep. No, you're good.
I cut you off. I was just going to say that I put in my email address in the chat in case people want to reach out. And then I'll stop talking.
No, perfect. That's great. So yeah, her contact information's in the chat. So feel free to ask her any additional questions about her work. And with that, have a great rest of the day, everybody. And thank you, Anoushka, so much again for your time.
Of course, thank you. And thanks everyone for attending.
Take care.